which was about this word, what if? This is being recorded, thank you. So when the pandemic hit in late 2019, one of the things that happened was that a lot of sources of information about the virus was opened up to the community. This was sequencing information, publications that came about from studies, and it begs the question, which is, do we need pandemics to open up data? So hopefully, through our journey today, we'll ask this question over and over again, and it's predicated on this perhaps an absurd question. I don't know if it's a hypothetical one. What if all the data in the world were instantly accessible? And when I say that, for the purpose of today's talk and today's discussion, I'll focus on perhaps neuroscientific or brain-related data, brain-associated data. I'm not going to mean every piece of data that is being generated everywhere, but let's focus on brain-related data. Okay. So now some of you may ask the question, is this even possible? And this might be utopian for many, but I'll start with the US side of things, and I'm sure many of you and Melanie and others might be able to tell me a little thing or two about initiatives elsewhere. So this is the strategic plan from NIH about data sharing and informatics infrastructure. So data infrastructure to optimize data with storage and security. They're obviously thinking broader than even the scope I laid out in front of you today. A modernized data ecosystem. This has to connect with all the tools that we have, the compute tools. Data management analytics to support useful, generalizable, and accessible tools and workflows. Improve discovery and cataloging of resources. Training, workforce development, which is partly why many of you are here. And then in the long run, how do we maintain, what do we preserve off of this data? Can we store all the data ad infinitum and things like that? So how do we steward the data? How do we improve quality of the data so that it becomes useful over time? And this is a plan, it's a strategic plan, and they've set it into motion. So as of grant proposals going in January of 2023, anybody who receives an NIH grant will be required to share their data as soon as possible and no later than the time of an associated publication or the end of the award or support period, whichever comes first. And that's a fairly stringent policy. Now, the other thing that comes with this is they have defined what they consider scientific data, the kinds of data management elements that would be required to come into play as you write these plans, where you're going to share the data, the metadata that's associated with it. So they've kind of thought through many different components of data sharing that needs to come into existence. So I know there was a question earlier today, how do I get somebody who's not inclined to share data? Well, there'll be something like this coming, whether they like it or not, at some point in time in the next couple of years. But hopefully we have a cultural shift that's happening independent of this policy. And I'd like to, yes. Good question. I don't know the answer to it yet. Where I know it has teeth are some of the consortial pieces that I will talk about a little bit that are already in existence. So some of the initiatives at NIH, particularly the BRAIN Initiative and the National Institute of Mental Health, which might support many of at least the US grants and folks in this room, already have pieces of this in existence, and it has teeth in the sense that award letters go out only if you've shown some of these things have been happening. So in that sense, there's some teeth. How they're going to do it at scale, I don't know the answer to it, and that would be a question for NIH policymakers and deciders. The other piece of this is a program like this. Now, this hasn't started yet, but this RFA or this call was announced last year. It's called Bridge to AI. And the intent of this is to generate data. So this was another question that came up. Who's going to generate the data if we only need large data sets? So there are some specific programs that are coming into existence about generation of data. And with the intent that these data are generated paying attention to kind of ethical concerns, privacy, security, distribution, scale in relation to machine learning and AI problems. Now, this is not all of science. This is going to focus on curating data sets for problems that lend themselves to AI and ML approaches. We will see when the awards come out, what kinds of awards they are. But this is another way in which NIH is helping support that generation of data. And they're doing it in a way that allows for a group of people or a consortia of people to come together and say, this is what's needed to generate the data. So Russ mentioned the ABCD project as one example of how it brought together a group of people to kind of come together. And they had, there are always pluses and minuses when you come to generating data at this scale. So the ABCD project is ongoing still. The HBCD project, which is the precursor or the prequel to the ABCD. So if you've watched Star Wars, we've done episode 4, 5, and 6, or 4 and 5 have happened. Episode 1 is starting, so they're going to start collecting data from the second trimester of a mom and follow the mom and the kid for 9 years. And that's going to start data collection this fall. So there'll be a lot of projects out there producing a lot of data for people to look at. And again, it may not impact the immediate grad student who's trying to graduate, but at least these are long-term initiatives that are trying to generate data at scale to allow for different kinds of questions to be asked. Let me flip over from kind of the policy side and the data generation side to technology. IPFS simply stands for the Interplanetary File System. And the intent of this is to make data accessible at scale across the web easily. So this is a technology piece. It's been in production for about 10 years at this point, and I would still say it's in its infancy in some ways. But even in its infancy, it has managed to share through something called the Filecoin network about 130 petabytes of data. Now, for those of you who have a laptop in front of you, think about 130 petabytes of data. Now, this is, again, not neuroscientific data. We're not talking about this, but this brings up questions for us. We have this available. Somebody is sharing some data. What do we do with it? Can we do something with it? And those are the questions I'd like to kind of bring up. It's like, what if the data was instantly accessible? The other source, much closer to home, and this is the pattern I'll try to repeat. I'm going to go through a few vignettes today. And for each vignette, I'll try to present a broader ecosystem of things and then try to bring in something closer to us. So DataLad, which Adina will talk about in a few days from now, or some aspects of it, already shares about 500 terabytes of data. And this doesn't even come to bear some of the other resources like the UK Biobank, the ABCD dataset, which cannot be shared openly through DataLad, right? We already have access to scales of data that we may not know how to deal with or do things with. So here is my answer to my question. If it became instantly accessible, it would be mostly useless. I'd love to hear your thoughts on it before we move further. And you can use the channel, the Slack channel, to type in, and I'll read things out. Or you can say it over here, and I'll repeat it for our Zoom audience. Yes?
I guess I'm wondering, in this scenario, would they be accessible from a centralized place?
Okay, so accessibility, right? In the sense of, can I get to it? So I don't know about central. I just talked about IPFS as a decentralized mechanism to get at things. But it might still be the case that moving these 400 terabytes of data is not going to be chump change for most people, or even storing it. So there are questions that come about even if we had access to data. Okay, so let's say it is. I'll take it's all centrally accessible. And what happens behind the scenes, I don't care, but you can go to one place to find all of these things. Yes?
An issue that you have all the data, like it must have requirement I together might be really hard, like PDF that might have they might be talking about the same thing, but like the thing that's different with like knowledge centers might be different. So you don't have to know how to combine the data if you want to do this analysis.
So for all of you, how many people here have tried to download data from someplace for your use? Okay, that's a significant chunk of this. I'm sure you've run into what Rahul just mentioned about interpreting the data that comes through. And I know there was a question earlier about the kinds of experiments being slightly different across people. How do you integrate different pieces of data that might be related to the same question, but may have scanner differences, population differences? So we'll have to figure that out. Okay, so I'll give you some magical tool. There's a magical fairy that comes with the data for now, who will answer your questions. Okay, what else? So most of the data is not well described, and I'll just keep this. Oh, I'm sorry. Melanie says I'm supposed to repeat the questions. I did say that. So the first question was, are all the data going to be centrally accessible? So there are questions and thought processes around it, but for the moment, we'll assume that that exists. The second question was, is there some good annotation, or how do you integrate data when they might have different ways of annotating different kinds of pieces? And I think that's also related to the third question, is how do I get a description of the data? So beyond the CSV, but can I get more information about the data? And that's the third question. So we'll need that. So that has to be available. So for now, we'll give it, right? Anything you ask for, we will give it. Yes. You need a question to answer. And that, I would say, is a very important question, right? I mean, there is all the data over here. We must have some notion of questions to come to this data with. So, okay, we'll need questions. And now let me see if I pick up some of the things. Is it because legacy neuroimaging data were all collected using different scan protocols? I wouldn't say legacy. I would say even current neuroimaging data is collected with different scan protocols. We are not at the point in time where every person collecting new imaging data is using the same exact protocol. There are classes of protocols, perhaps, but there's still differences in protocols as data are collected. Okay. So I'm still on the optimistic side, so I'm going to give you something else. This should become more easier to do if we had access to data. Now, subject to all the things that I have given you. You have access, you have metadata, and you have a question. So this kind of brings together this notion of a question. I've done some analysis on some original data set with some scripts. I wanted to see if it is reproducible, right? And I've broken down reproducibility into kind of this 4 quadrants of re-execution, replicability, robustness, and full generalizability. And I think this is a useful way of breaking it down, because when we use the term reproducibility, a lot of us kind of mean it in different ways. So we're just kind of giving more concrete pieces to it. So the original data set with original data analysis is, can I just repeat the same thing that I've done? I have my data, I have my scripts, can I hit go? And it returns the same kinds of results, perhaps within some statistical limits, because you might have random number generators or other things within that step. The second one, similar data set. I might need to find one or need to collect one that is similar. So having access to data is going to increase the likelihood that I might find a similar data set. So it's going to help with replicability a little bit. Whether it's exact, similar, those are questions that we'll have to think about. What do I mean by 2 data sets being similar? Are scan protocol differences okay? Are participant differences okay? Are time of day differences okay? It might depend on the questions we are trying to ask and answer. Robustness. So this, I keep the original data set, I just improve. This is similar to the NARP study that Russ talked about earlier. I just create different kinds of analytical pipelines that are supposed to be similar, so I can look at how similar my results are across these things. And then full generalization kind of says, okay, I did something. I should be able to collect something similar and work with another similar pipeline and still get the same outcome. So hopefully having access to data would help with that. And then Adina reminded me yesterday on the ML side of things, This probably comes into play, maybe not just on the ML side of things. So we talked about, or Russ talked about, analytical flexibility in the morning. Perhaps data flexibility has as much of an issue at a certain level as analytical flexibility. You could find some piece of data that matches your hypothesis. This is a little more drastic, but I think the same kind of principle holds. So the other thing I would say is people have had access to data for millennia, whether they were looking at stars systems with their naked eyes, or whether they saw things drop from a tree down, or any other thing that dropped. However, for the latter case, it took a lot of thought process and evolution of scientific understanding to go from, I can observe something drop, to here's a model of how two bodies interact with each other. So data has to come with, I think, questions and models as well that kind of bring this together. Further, for those of you who have seen this beautiful image, this came out just about a week and a half back. This is from the James Webb Space Telescope. So we had been looking at stars, but we needed to look further in time in this particular case, and with higher resolution. So we had specific questions that we wanted to answer, for which we needed to build new tools to get at new data. And I think this is important to kind of think about, which is that scientific questions often need specific data and tools. So even having accessibility solves some bits of the problem, but you might have a question for which, even if the fairy was there to answer whether there was data that existed and was accessible, you might not find something that answers your question. So you might have to go ahead and collect it. And in this vein, I'll give a couple of examples within my own group. So some of my group does work on understanding the neurophysiology of human communication and its translational applications. So we look at how we speak and how we communicate with language. It's easy to acquire today. I'm speaking, everybody on Zoom is listening to me over my microphone. You are listening to me, and there's a process in play. And that same equation that related to the understanding of why that apple drops on the floor is also related to those things we call F1, F2, F3, which are kind of spectral resonances in the vocal tract. So it requires not just data, but an understanding and a model, excuse me, to get at understanding a system in place. Now we can take the system, do some machine learning, and we can try and dissociate people with different levels of Parkinson, for example. And we can show that certain analysis of this data will give rise to certain markers of whether you have mild, moderate, or high Parkinson's. Having more data in the space would help. I'd be able to verify, validate this with greater spectra than what we had in this particular analysis. Similarly, we can take some of these things, and phonemes are kind of segments of units of sounds that we make, and relate it to things like depression level. And we can then take that, have people do this in a scanner, and we can then say that while you're producing speech in the scanner, the activity in certain parts of the brain are related to your depression severity. Now, if I go to my fairy and say, give me this data so that I don't need to collect this, that would be hard for me to get at, because that narrows down the set of things that I need to answer that question or create that plot. And that's what makes data kind of challenging. We're not asking for just broad strokes of data. We are asking specific questions around certain types or facets of it. And those dimensions make it harder to get at the right piece of data. In a similar vein, we looked at the literature and said, okay, all the people who have done analysis on voice in mental health disorders and encoded kind of the different acoustic features that come from voice with studies that have been done across mental health disorders. So having access to more data might help fill out this space. So anything that's blank means we could not find a study in our search at that point when we did this. Anything that is colored tells us kind of the level of support of that feature in its relation to that disorder. And the size of the box kind of tells us how many studies, roughly speaking, are supporting that piece of evidence. So anything that's tiny means maybe there's a study or two. Anything that's large means there are more studies that have been done looking at that acoustic feature in the context of the disorder. But as you can see, this landscape is fairly empty for most part. And having access to voice data across mental health disorders would help fill up some of these things. Let me shift gear to a different project that we've been doing. So we built a tool to help people who stutter less. So increase the fluency in people. And all it does is it picks up the voice as somebody speaking, sends it to a phone, which does some processing, and in real time sends it back to the ear. So it's like a real-time system. I'm speaking and it's altering my sounds in a certain way, and I'm listening to this. And when I do that, let's see if I can. Oh, magic might happen.
Had speech therapy since Harch, the person I'm working with, is like she specializes in she specializes in stuttering.
And when we turn this thing on, this is what the same person.
The Notes app I use all the time because if I think of something, I'm going to forget it, so just write it down. Or like little things that I see that I want to look into later, I write it down.
Okay, so get my mouse back. So something like this was built to kind of create a low cognitive load, inexpensive, reduces habituation, a more pleasant thing. People can actually listen to this kind of approach. But even if all the data in the world were accessible, would I be able to find the pieces that would have helped me develop this specific app? Where would I have found it? What might I have? I might have gotten access to certain kinds of people with stuttering in all of the data that's being collected. But coming up with this app requires more than just that. And so this is my question: is instant accessibility data enough? Now, these examples I gave required kind of knowledge of computational neuroscience, neuroimaging and cognitive science, psychiatry, speech language pathology, machine learning, signal processing, acoustic phonetics, linguistics, software engineering, infrastructure to come into play. So just accessibility, I would say, is not going to cut it for us. So I'm going to stop over here, and just based so far on what we've covered, why don't you either in the Slack channel or over here kind of think about or say what other things you might need. So this speaks to a lot of training. And there was a question earlier this morning about what do I have to learn to do science today? And there are many pieces of this. So what I want you to think about is, as you're doing science right this minute, what would you change, a thing or two, that might make it easier to solve some of the problems we have seen thus far? And how hard would it be, and you can be honest over here, to implement it in practice? So we'll leave education aside. All of you do learn a lot in various places. You're here. But what are the things that you could do in your own practice to make data not just accessible, but usable? Yes. It's something that you might want to do for a publication anyway, right? And if you, oh, sorry, the question, thank you, Ariel. The question was, or the comment was that you can get detailed information about how you collected the data, participant inclusion, exclusion criteria, and encode it in some way that you can distribute together with your data, or even have it with you as you give it to the next person in the lab who might use it. So you could be in a very, very selfish mode of doing this and still be helpful to other people. Yes. Fantastic. So protocol papers. So the comment was that we don't have enough space in the methods section to put in all the details. We can write protocol papers and make those accessible. And in fact, I would say there's a place called protocols.io, which exists, and you can put any of your protocols of any kind, whether it's data collection, data processing, or others in it. It's just very similar to your bioarchive or your archive preprint servers. You can get a DOI for your protocols. You can even cite it in the method section as here are the details of things. Yes.
There are many types of them having their own pattern. So it would be really good to have a pattern way for describing data and describing protocols and everything that we understand data.
So the comment was that in addition to kind of describing things, it would be nice to have standards for describing things. And hopefully I'll touch on that a little bit in just a little bit of time. Yes. So the comment was that it would be nice to have a best practices guide for various kinds of things. So we could, both understand problems that other people have run into, but also use it to kind of guide your own space of things. All of these would be fantastic. In fact, there's an OHBM best practices committee. So if you think of things that really should be encoded, you should write to the committee and say, it would be really nice to have this. And as a society, and that's where the collaborative part of this comes in, right? The reason I listed this is not one of us can do all of these things as a person. We kind of need a group of people to collaborate with and be able to do these things. And so maybe one of the things is, if you know that there are other people doing similar things, reach out to them and say, can we work together? Because that might empower us to collect more data, because each person has limited resources. It might allow you to think about coordinating best practices across multiple things. And this is something that you can do at a grassroots level, in addition to some of the consortial things that are happening from bodies like NIH. Okay. And I know Russ mentioned this this morning, so I'm just going to put it up there. So one such example is this consortium called Enigma, and they've been focused on getting many people together because their original thread was looking at genetics and brain imaging information together. And since you need large numbers to kind of look at gene-brain associations or other things, they knew that no individual lab would have enough capacity to both generate, I mean, they might have capacity to generate a large number of genetic sequencing data, although when Enigma started, genetic sequencing was still pretty expensive at that point in time, but let alone the combination, right? Gene-brain associations. So they started bringing people together in groups, studying specific disorders and saying, you're interested in gene depression relationship, come together and let's take all the labs which are doing analysis of psychiatric disorders of various kinds, form a working group, bring data together from their individual sites. Now, they focus for most part on structural images because those were easy to access, and genetic information. Today, our questions have become increasingly more complex, but the same principle of kind of coming together could hold towards more complex questions. And that reminds me of this tweet. This was, I think, about a month back. This was a paper that was published with 8,778 authors. I've not reached out to one of them to ask if they adjusted it to make 8778 a palindrome or not, but I think part of this reflects the notion that groups of people can come together towards solving certain problems And there's a way to acknowledge that all of these people participated in this. This is not about credit in the sense of first authorship or last authorship. This is simply says, this was a complex problem. There were lots of people involved, and they came together to solve this problem together. And some of this is happening in neuroscience as well. So this is an example of a paper that had about, I think, 250 or 300 authors, where they went across species to look at motor cortex and relations in cells of motor cortex across those species, from transcriptomic, neurophysiological, morphological, and other properties. And this was part of this consortium called the Brain Initiative Cell Census Network that brought together these people to work together. They also published their individual papers, but this was kind of a flagship paper that summarized and integrated a lot of those individual results into one. And that brings me to my second vignette. Neuroscience is complex. We have to look at all kinds of tools, resources to truly put together a picture of the brain. Neuroimaging happens to be one of them. But to understand how the brain works, we need to look at it at multi-scale. We need to have theories, models, and as well as experimental designs. We might have different driving biological applications. Some of them might be clinical and behavioral. Some of them might just be knowledge generation. We don't know enough about something, and we want to know more about it. We need to build new tools because we might want to see further, like the James Webb Space Telescope. We might want to see farther into the axon, the molecular processes. We can't do that at this point in time very easily, but we might want to do so. And we need data at different scales with rich descriptors and provenance. So many of the things that people brought up here is this notion of provenance. Do I know what happened to the data to get there? So accessibility by itself will not solve this problem. But one of the things that I think is happening is we have a lot of sources of data today. So this doesn't even have to be a problem of the future. This is something that you can start with right this minute. You have OpenNeuro, the UK Biobank, and these are a list of all the Brain Initiative data archives that have been funded by the NIH, making data publicly accessible across these different scales of neuroscience for you to look at. Bing will be talking in the next session, and I'm sure she'll be talking about some of the data that's related to neurophysiology as one of the things that's stored in one of these archives. But this is coming online, and they're doing so in somewhat of a standardized manner. This is not your Figshare or your Google Drive or your Dropbox dumps of data. The intent behind many of these archives, and OpenNeuro is one probably many of you are familiar with over here. It uses BIDS as a standard to kind of describe data that makes the communication of data easier. Is it 100% that you don't have to contact the authors? Perhaps not. But that's a process that, again, many of you can participate in or in your own work. When you say, I can encode things better, I'll ask you the following question. And I say that, so Dandy is one of the archives we run, and I'll talk a little bit about that in a second. I kind of ask the question. So NIH is going to make most of you, or many of you, put data in these archives. Could you put data in such a way that if fire hosed out or some computer accident hosed out the data that you had locally, that you had encoded the data that you put on the archive with sufficient information for you to do your own research? I'm not asking you to do this for anybody else, just for yourself. Do you have enough descriptions that you 3 months from now or 6 months from now could look back on your data and say, I know exactly what I did to this or how I derived this, and it's encoded there. And if you could take just those little steps for yourself, you're going to also make it much easier on the community as you do that. So perhaps that's a step that you can take. Think about the details that you left at the scanner table when you walked away from it. Could you have encoded it in some structured format while you were doing that? So this has resulted in, and this number is already a little old at this point in time, about 100K brain scans that we have access to at this point in time. And one of the challenges that has come with this is they come with varying quality. So we have had to build a few tools to kind of address this. So one of these tools is this tool we call Quick, which runs on T1. It doesn't evaluate every single type of brain scan, at least yet, but it can take T1 weighted structural images and give us some number that is an approximation of its quality. And we can use this and run it on something like the UK Biobank, which has a couple of labels associated with each data that it distributes. And this was manually done to say usable and unusable. And the process involved a fairly complex pipeline, but one of them was whether it would run through a few specific FSL tools. So if it failed that, it would be called unusable. If it passed that and the other criteria, it would be called usable. So we ran Quick on this, and one of the things Quick generates, which is in this bottom row, is a notion of uncertainty. It's a Bayesian neural network that tells me how sure it was as it was solving segmentation problem in the brain. And it's this uncertainty map that gives me a sense of the quality. And if you look at just these 4 columns, you'll notice that the first and third column, at least on that bottom row, says uncertainty is high. And if you look at the original images, you will see a lot of movement in those brains. If you look at the second and fourth column, uncertainty is low, and they look clear to us. And when you looked at it and evaluated as a neuroimager, the brains look good. So this was a way of saying there are tools that we can build that evaluate certain things. It still has assumptions built in, but it might allow us to kind of scale this across large data sets. So we did. We ran this on OpenNeuro. So each column over here is a data set from OpenNeuro. And we generated an uncertainty value for every brain. And so you're looking at a box plot where each column is a data set and each distribution is this uncertainty measure from the data set. And we put this magical number called 0.4. It is truly magical at this point in time. It's as if a fairy came down and gave it to us and said, use this threshold. Anything above it is bad. Anything below this is good. We don't know why yet, but it seems to work it's the empirical side of science. And if I look at the data sets over there, we get to see some of the reasons why I thought it was bad. Might not have been a human data set, might have been a partial brain, might have been a quantitative T1 image as opposed to a T1 weighted image, even though it was labeled a T1 weighted image. And we can look at a couple of the ones that are on this side. And now again, you can see that they had movement in them and they got picked up. So overall, this data set is pretty good, but there are some that are higher than that magical number. And when you look at it, you either see partial coverage, motion, or both together. So these are the kinds of tools that having access to data would help us build and we would need, because we won't be able to go through 100,000 brain scans manually. But it might also be the kind of thing that we could crowdsource and kind of think about how you could input your notions of quality control into this. But what is our notions of quality control? We don't know quite yet. And this is one of the reasons we've kind of started this effort. So this is a Jupyter book, which is a re-executable publication of sorts, where we're starting to put together kind of different notions of quality control in a book. And the idea is that this is a completely open book. So if you have quality control protocols in your lab that you wanted to document, you might have that for infant imaging, as an example. You might have that for, here is how I do quality control on a T1 weighted image. You can start contributing to this. It's a chapter. You can contribute a chapter. This is my lab's quality control protocol. And as we get this from different places, perhaps we can reach that consensus on what quality control might mean across different types of things. Because quality truly is in the eyes of the beholder in neuroimaging at this point in time in many cases. Okay. This is the other side of the thing, which is, this is like a general notion of how people do computation on various things. So this is something that we're doing as part of the ReproNim project, and ReproNim stands for a Center for Reproducible Neuroimaging Computation. So you have your usual kind of data analysis compute thing, and then you store kind of metadata and provenance in local data stores, and then you expose some of it because we are dealing with human data. You may not be able to share everything publicly. So you can share some of the things publicly in public stores with the notion that they still have connections to the local stores where you might be able to get access to things. And these access models will change over time. We're not quite there yet. If you've dealt with UK Biobank or ABCD or any of the NDA data sets, you know you can get access to more information, but you have to go through steps to get at that information. But when we say standards, we're talking about some of these standards. They come in different flavors, right? Here is the Hcp Reference Manual, which is all in PDF. It's descriptions. It's rich, but it's all in PDF that you have to read. Then there is the brain imaging data structure, or BIDS, and it's all structured, but it might still miss some of the things. We might not be able to know how the data came to be in that place. Those things that you might take in lab notebooks might not get there. So one of the things that you might want to do is to extend some of these things with richer semantic annotation, which is where the neuroimaging data model kind of comes in to say, not only do I put things in, I know exactly what I mean when I put these things in, which ultimately means that there's a common vocabulary we all share when we talk about scientific data. But we can't, magically come up with a common vocabulary, because even when I say a T1 image, somebody might have their ideas of what a T1 image is. And this is where efforts like NIDM terms and others kind of come in to get communities to build common vocabularies and share each other's terms so that we can be consistent about how we communicate. We all talk in English, I know. You're all listening to me in English, but English is a very flexible language, and scientific representation in English comes with a lot of flexibility around what it means. So could we be more specific about the data that we are sharing? Could we be more specific about the kinds of elements that we are describing? And this is where, again, as you do things, you might be able to say, I'm studying this niche thing over here. Here's a term that I use. You might go to one of these communities and say, I'm going to add my term. It may not be used anywhere else, but it's my term. I'm defining it this way, and this is how I've put it there. It opens up the discourse, which will help people who then get your data set with that term precisely know what they were looking into. And we can do some of these things for a class of data that we don't think about a whole lot, are form-based data. So here's kind of an ontological tree of describing forms-based data that we might collect. So protocol. And activity might be something like the patient health questionnaire. It's a set of questions. So this is an activity you're asking people to do, answer these questions. The questions themselves are fields that you can look at. You can encode how they're supposed to respond to it in response options. And for those of you who have used REDCap or other such tools, this is kind of similar ideas. But think of the REDCap instruments that you have used. They're all idiosyncratic across labs. So when the variables come out from it, unless you're forced to organize it, you're not going to make it there, right? So this is why we developed this thing called repro schema, which allows people to kind of get these things into a common public space and reuse it in different places. So you could export this out of repro schema into your REDCap system and use it. So we're not saying get out of REDCap. We're simply saying use the same kind of variable names that everybody could use together. And if you are going to add a new form, add it to this library of forms so that other people can reuse those questions and know specifically what questions were used. And then we can take responses and store that as well. And so all of this can be connected through provenance down into some structured format. So we may not like structured formats. It sounds too computer science-y. I don't want to do it. I prefer writing Word docs or Google Docs. But if you could take a little bit of a step towards either creating them or reusing them, you might find in the longer term, they really carry a lot of potential for you to reuse your data and to reuse other people's data, because now you know exactly how it was created. So I'll come back to that question. Is instant accessibility to data enough? And we'll pause again. We have a little bit of time, right? Yes. And So there are issues around quality, right? Just having data on its own, you'll still have to know, is this good quality data, bad quality data? You might need tools around it. You'll need to evaluate it somehow. Metadata and provenance. You have to have information about how the data originated. And this could be at all the levels of data that you might think about, right? Raw data from the scanner. I need to know the experimental design. I need to know why this was collected in the first place, who it was collected from. If it's process data, what processing went on, which versions of tools were used, what atlases were used if you're doing neuroimaging analysis. And that's provenance. Those are details that often get skipped out in some of the method sections and other things. Filtering. Okay, we have all of this data, or at least in our hypothetical space. How do I filter for things? How do I find things? How do I search things? It's already hard, I would say, even with the data that's currently available. How do you look for it? We'll still have that problem. And then there's human resources. A lot of the things we've talked about often require human things. So how do we flip this around? So I'm going to pause again and say, you're doing things in your lab or in your work. How do you step back? And what kinds of things could you do as you are playing with data that would make it beneficial to you as well as to others, so that we don't need as much human resources on the other side? Could you do more consistent quality control and publish your quality control protocol, for example? Could you, and that's going to be limited to the data you're playing with, right? If you've collected, presumably it's not at the 1,000-person scale. It's in the more 50-ish to 100-person scale. That's a scale that's manageable for you. Could you generate a brain mask for every single person so that there's kind of a ground truth brain mask that other people could reuse? And it might help you in your own processing as well, because you have a golden brain mask there. Could you annotate data in a way that kind of links that data set to the thought process that went into designing it? So I'm going to stop there and say, okay, what could you do? I've suggested a few things. Let me flip the question around. What are the things that trouble you when you use data that you could avoid by doing things yourself? So this tells me that I can look at the distributions of things in a certain way, and those are annotated alongside the data. So if it's brain imaging data, sure, I can look at image-based distributions, but I would need to also look at other facets of data collection, maybe gender, maybe ethnicity, maybe age. I bring up time of scan, kinds of things, but those have to be encoded there for me to look at those distributions. But yes, I think understanding distributions of data can be quite useful when you're looking at large scales of data, right? You might find, hey, there's a single kind of Gaussian or single modal distribution of this data or this dimension, or I can see, 3 peaks in this. And so I know they come from different kinds of distribution. So being able to visualize those things is going to be useful at scales of data we are looking at. So 100,000 scans. If you're looking at, okay, I'll take something again close to some of the things we do. Heschel's gyrus. So for those who are unfamiliar with that term, it's primary auditory cortex. And some people have 2 humps and some people have 1 hump. I would love to know what that distribution is in those 100K scans that I'm looking at, right? And I would need to be able to, okay, I can't look at every one of them individually, so I'll need some tools to process it, which requires going back and ensuring that I have T1 weighted scans that I can look at the, so I might be able to even generate new distributions off of this that might help me do further analysis. That depends on that. But I'll need to do some kind of automation, and hopefully over the next few days, you'll learn tools that help you scale your processes to larger levels. Okay. So any other things that you have trouble with when you download, or data is hunky-dory? Any data set you download, it's like fantastic. I can go with this.
I have a quick question.
Yes.
I'm on Zoom, but I think maybe I've done more clinical research than basic science. So from that, I feel like most of the studies that I've done always try to control match for age, sex, or some sort of qualitative measure, like in stroke would be NHS score or something like that, and then analyze the data after viewing the distribution. So that pipeline seems to be the best way to look at something more acutely. It may not be as efficient in certain respects, but in what you were saying in filtering, is that more of a step one, step two, or is it that it becomes evident as you view the data? I'm not sure.
Okay, so from that perspective, let me flip that question around differently. Let's say you were looking for stroke data and you went to Open Neuro. How much stroke data can you find on Open Neuro? And what would have it required for you to find that data? I would think that somebody would have to say this person had stroke, right? But you might want other things like, okay, did this image get collected before this person had stroke or after this person had stroke? So I think that's where that filtering question or annotation question kind of comes in. You're looking for different dimensions associated with data. And unless those were annotated in a way, and so in our group of people talking about annotation of disease and diagnosis, we asked the question, so if somebody said this is a controlled subject, what does it mean? Is that a neurotypical subject? Was that a control for a particular type of question? How do I kind of decide that when I look at open data sets? And that's, I think, what I'm trying to get at is you need richer annotation of information to determine certain facets. And if you can do that, because when you do the study, you are encoding, you are deciding what that inclusion exclusion criteria are that you have used. Can you include that when you send out that data set? Or I know it's in publications that eventually come out, but it's not always the case that every data set will have publications. And I think that's going to be more true as we move into the next few years. But speaking to your point, and I don't know who it was because I can't see things, I think you're absolutely right, which is you are looking at these distributions to match, but you need to have information in the first place to get at those distributions. Yes. I would say that's better than our current state. I'll take that growth. Hopefully it comes with some kind of standardization around the terms that are being used to annotate these things. It's not just free text annotation, I'm hoping. And that we understand kind of, if you're looking at diagnosis, maybe there's an ICD-9 or ICD-10 code that comes with it. So I would stay away from free text to the extent possible, but at least even free text is better than no text at all when it comes to annotation. But hopefully we can all do better in getting kind of structured text into the space. Right. Let's continue. All right. What scales of data are we looking at? So I already brought up this table earlier, but I'm going to flip it around to some of the things we do in our group. So we are interested in kind of predicting the future of us in various ways. We want to know if, we'll be having a depressive episode or we'll be having better sleep. And so some of the things we are doing is measuring these things out of various different sources of information. And it could be objective measures like brain scans, wearables, and other things. Could be subjective measures, people filling in those forms and doing things. Or could be information about interventions that they've participated in. So we're interested in kind of looking at trajectories of people and understanding how those trajectories might change, what perturbs the trajectory, what assesses different components, and hopefully we can toss this into, I have hated using the term AI assistant there when I first put it there, some sort of information processing system that amasses these things and tells us in a predictive manner what the future of this person is going to be. So then we started looking at all these things that we collect. Our whole genome is about 300 gigabytes. MRI is small by that standard, 3 gigabytes per hour. MEG, EEG 2 gigabytes per hour, video 5 gigabytes per hour, microscopy ex vivo, it's only about 300 terabytes. So we've now jumped a few orders of magnitude in this space. And this brings us to the space that as we collect data, whether it's scales of people or the types of data within a person, we're getting to more longitudinal scans. We might get at scans, pre-mortem and then post-mortem. So the scale and scope of neuroscience is changing. We're also getting probes like the NeuroPixels, which used to generate about a terabyte in 13 hours. Then they came up with version 2. And this increased the number of recording sites to about 10 times. And they're working on NeuroPixels NXT. This is kind of in development. And the story I hear is this will generate about 0.5 petabyte of data per postdoc/grad student's experiment in a year. We aren't used to this scale of data, but we have to think about not just the annotations around it, but the tools, the infrastructure that supports some of these scales of data. So one of the projects that I've been involved with is DANDY, which stands for Distributed Archives for Neurophysiology Data Integration. And we store about 400 terabytes of data, of which about 320 to 330 terabytes is microscopy data. It really is the largest kind of segment of data that comes into it. We're using standards. We use Neurodata Without Borders and BIDS to store information. We have a Jupyter Hub. So as you get to learn about the Jupyter Hub over here, as you use it, you'll see the kind of flexibility that that kind of environment provides, especially in relation to this kind of scale of data, where you're sitting with your compute right next to your data. You've always done that, whether it was in your laptop or whether it was in your compute cluster or now in the cloud. But the cloud just allows a level of horizontal scalability. And I'll show you an example in a second where any of you here and on Zoom and elsewhere can access some of this data simultaneously without me having to think about, oh, did my bandwidth get used up as you were accessing this data? And that's the kind of scale that these data archives are hoping to provide to people. Okay. So here's an example of a data set. This comes from Bruce Fischel's group over at MGH. It's a data set for ex vivo scanning. They do MR at MGH. Then it goes over to BU, and they do block face OCT, optical computed tomography, which slices this ex vivo slab and ships off those slices over to Lens in Italy, in Florence, where they do light sheet imaging on it. And then the digital versions of this go over to Columbia University, where they manually look at sections of this and count cells. Hopefully to build up future versions of analytics that can do this more automatically. But at present, this is the pipeline. And so what this infrastructure allowed them to do was to feed in data from multiple places in standardized form into a central archive, so that each of them could upload their pieces. Now you could say we can do this in Google Drive or Dropbox. And at this scale of 11 terabytes, probably you could, although I don't know how many people have access to 11 terabytes of space, even on Google Drive and Dropbox at this point in time. That's getting to a range where it's getting complicated. So let me shift over to the next data set. So this data set is a single human hemisphere imaged every millimeter through clearing, and then imaged with 3 kinds of stains to look at distributions of proteomic markers in the tissue. Now, this is not your typical neuroimaging scan data set, but this is a kind of data set that gives us information about distribution of cellular information in a human. And this is a unique data set because there are not too many of these kinds. Now, this data set currently sits at 320, and this I downloaded. This snapshot was taken May 19th. I think it may have another 7 or 8 terabytes by now. But when you're talking about this kind of scale, you're not going to download all the data, right? You have to be able to bring your compute as close to this data as possible. And this was a challenge for this group and this community. So we worked with the BIDS community and the open microscopy community. And this is where it's important. It's not individual things. We're dealing with challenges that require communities to kind of come together. And for almost anything we do, we kind of see that the community-based interaction helps us solve a lot of problems. So you're all pretty familiar with BIDS, I think, over here. It provides a uniform representation of diverse modalities and related metadata. The open microscopy community was doing this for microscopy slides, digital pathology, microscopy, all kinds of things that require taking photographs of tissues or other things. And they had been moving with this format called OME-TIFF, which allows people to store metadata in addition to. TIFF is probably a format most of you are familiar with. It's an image format. It can store 2D or 3D or more D volume. And they moved to this other format called the next generation file format. And this is a ZAR-based format, which can store petavoxel scales of data, can have metadata associated with it, and can store encoding information about transformations. What we did was we brought these two communities together and helped create an extension to BIDS, which allows storing of microscopy data alongside BIDS data, because we are going to see much more data sets coming online that have some component of microscopy alongside brain images being generated. And this helped us kind of store this data set that can be viewed by anyone and everyone in the cloud and accessible using multi-scale representation. So if you wanted to look at the coarses resolution of this data, you could do so. You can have information about scales and transforms. And we can use tools, and in this case, we're using NeuroGlancer, which is a tool that a group at Google has produced to be able to view this directly. And they put in the support for OME-ZAR into NeuroGlancer. So we were able to stitch these pieces together to kind of give people access to 300 terabytes of data that they could visualize on the fly. We could then build dashboards like this, which tells us the quality of data for each sample that was uploaded. And we're kind of color coding it by different kinds of errors that were detected by some of our tools in these samples. And this is what becomes possible. Now, when we think about imaging data, we might say, okay, we're not quite at that scale. So let me take a step back. How many people here do fMRI? A lot of you. How many people have generated a dense connectome? Okay, that limited my question to a few people. Okay, let me take a step back. You have vertices on a brain. How many people, all of you familiar with free surfaces, brains? So there's something called the FSLR template, which has about 91,000 vertices. So if you generate a dense connectome, which is, let's say for functional resting state data, the connectivity of one of those vertices to another, you get a 91,000 by 91,000 matrix, right? If you're storing that as float, which is 8 bytes, it would roughly translate to about 64 gigabytes of data. How many subjects in the Human Connectome project? About 1,200. 64 times 1,200 is about 72 terabytes of data. And this is the one reason they don't distribute the dense connectome for an individual out of the Human Connectome project data set. They generate an average dense connectome that they distribute, but not the dense connectome per individual. The Human Connectome project also has 4 resting state runs. So you might consider generating 4 dense connectomes. So 4 times 72, roughly speaking, is about 300 terabytes of data. So even in the neuroimaging space of things that you deal with, you're not too far from this scale of data. And so thinking about how to access these things is things as a community we should be discussing and getting to. And that's the Human Connectome project at 1,200 subjects. If you think about ABCD with 11,000 subjects, now you can do the math yourself to try and say, what would the dense connectome representation look like for that? And does everybody need to recompute it everywhere? And those are the trade-offs that you'll have to think about, which is, do I recompute? Do I store it and disseminate it? How useful is it There's a lot that goes into the computation of that dense connectome. There are pre-processing steps and other things. People may want to adjust it. So where do you store these things? How do you play with it? That's a scale of data you want to think about. Now, I'm not going to kind of show it, but this was what we have put up there is an example of an ex vivo data set that's stored in NIfTY as well as this ZAR format. And I can send a pointer in the Slack space later on around this. But the ZAR format allows us to view it instantly online. The NIfTY, because this is about a 3 GB file, I think it's a 3 GB file, yes, makes it difficult to visualize things online. You can download it. 3 GB is not much when I'm talking about terabytes of data. You can download it. But as you scale up to different things and you're getting higher resolution data, again, as a community, I think we need to think about what the formats and accessibility of these pieces are. So is instant accessibility to data enough? There are new technologies coming out pretty much. This is what neuroscientists live on, new ways of probing the brain. And so we're coming up with new technologies that look at different scales of data. And we're going to connect not just functional data, but all the way down to cellular and molecular data across. And that's the dream, right? Being able to do that, and hopefully in vivo. I don't know when that will happen, but that's a hope. There's this notion of perspective versus retrospective. There's data that's already available, but what can we do today for data that will be available that will make things easier downstream? Also, is the data that will be available swamp the data that has been available? As in, could we say we don't need the data because the next data set covers all the pieces of information that we could have generated from the past into it? Across neuroscience, that may be a little more of a question, because there might be species which turn extinct, and you might want to keep that old data around to do certain things with. There might be certain groups of people who might pass away, and you might want to keep some of that data around. So you'll have to think about kind of existence of data, or the life cycle of data a little bit. Scales of access and compute, whether you bring compute to data or how you access data. These are things we don't often think about when we are collecting 20 subjects worth of data in an experiment. But as we start collating these things, even the UK Biobank data set, if you just download the free surfer reconstructions, is about 10 to 20 terabytes of data. You need to have the capacity to store and process those things in various places. I've already covered this. So let me move on to the last section of this talk, which is, we've kind of talked about accessibility to data. The term FAIR, how many people here have never heard the word FAIR as an acronym? Okay, there's quite a few people over here. All right, so FAIR is now kind of written into almost every single NIH proposal announcement that comes out, and it's likely in every different country's proposal. So if you're going to be writing grants, you're going to see the word FAIR in various contexts. And it stands for findable, accessible, interoperable, and reusable. And this pertains to the information that you generate out of your scholarly research. Can it be found? Is it accessible? Does it work with other tools? Can it integrate with other data sets? Can it be reused for other things? Now, these are questions that you could ask of your own data. How would other people find it? And you could start with your lab and say, okay, can I make it findable within the lab to other people? Is it accessible? Now, some of these might be easier because of various setups, but these are things, especially if you're dealing with data that you've only used your laptop for. I think some of these questions are going to come up in terms of how do they get to this thing? And these are things that we don't maybe think about in the same way across different institutions, because we have different capabilities. We have access to different kinds of infrastructural pieces, but it's a simple thing to ask yourself for each of these data sets that you are working with. other piece to this, and this came out of kind of the Data Alliance, is care. We also have to think about who provided the data, who has authority over it, what can we do with that data, and whether this is related to GDPR in the EU, whether that's related to CCPR down south here in California, or in general about data stewardship. I think last week's Henrietta Lacks's genome was finally agreed to be used, or that sequencing line was finally reached an agreement for people to use. So those are the kinds of things that we have to think about as these data generate. And if you're collecting data, you want to think about the consent processes and other things that come around human data that you can relate to as you're collecting new data. So the brainconsent.org, I think that's the website name, contains a set of consent templates that you can use to guide your development of consent around data that you collect. And hopefully we can keep some of these things together in mind as we create data that speaks to the diversity of humanity in a way, as we ask questions and be able to answer questions in relation to whether it's, no, I'm not going to say this. I was going to say brain-wide associations because that came up earlier, and gets low effect sizes, or in general, kind of look at different kinds of relations to data that come out there. And I'll put this model out there as a model that people have been developing. It's this group called Open Humans, and the model is a personal data store. I put my data in a data store, and when somebody wants my data, they come and say, here's my approval to run this study, and I check off and say, you can have this data for this study. Now, in some ways, they've implemented it for certain types of communities, and I'll give a couple of examples of those. But in general, this is hard because this kind of brings with it all of those considerations that we talked about, right? Quality, accessibility, bringing compute, and other things. Most of the data they're dealing with is so far fairly limited in scale, and that's why they can use this model to kind of hold the data and distribute it. And one of the communities that has used this thing is the Nobism community, which is a community that deals with cluster headaches. And they've kind of used this model to help community members kind of come in. So as we are dealing with other types of health disorders, this is another way of thinking about it. You get invested people who are interested in understanding autism, or interested in understanding minimally verbal communication, or other things, to come together to say, okay, we are interested in learning more about ourselves. How do we come together? And this community has been using Open Humans to do so. Another community that has been doing something similar is the Open APS community, and this has to do with development of devices for automated insulin injection. So they're looking at diabetes, and the community came together and created one of the first closed loop systems for insulin delivery before something got approved by the FDA many, many years later. So there are things that one could do with communities when you involve the people who are producing the data to come together to work in the space of science. And I think that's something worthwhile to keep in mind as you deal with data. So I'm going to end on this note. There are many challenges. Curation and education is only the tip of the iceberg. When you're thinking about FAIR, thinking about standards, formats, and ontologies, well, they're tied to education, but you'll have to use and learn about these things, which means we need ways of creating technologies to make this simpler for all of us to do these things. And we're not there yet, but maybe an event like the NeuroHackademy can help you kind of bootstrap yourself with a few of these tools to get started. Provenance and knowledge, there are all kinds of lossy references in data out there. So if you think about yourself 5 years from now or 6 months from now, think through that lens and then code up your data. That extra little time when you are generating the data will be well worth it when you come back to look at it. And then at the scales that we are talking about, we'll have to think about sustainability and governance. we're not there as a community. We can't even standardize our processes yet. We think of 10 different pipelines to process our data, or get to that same kind of endpoint, a similar kind of endpoint. As we get to some of these scales, how do we decide what do we keep around, what do we throw away, and when do we throw away? And those are things that we'll have to get to as a community to say, okay, what is our consensus on fMRI pre-processing? Let's ask the question, why do we not have consensus on fMRI pre-processing? And those are questions, are hard questions, but we'll have to ask ourselves some of those questions one of these days. Okay. So this accessibility data is very much tied with fairness, and it's at the forefront of modern science. You have to have things findable, accessible, interoperable, and reusable. All of our science is getting increasingly more complex and multimodal. When we think of it, and this is what makes data hard to find, because we are asking questions that have different dimensions to it. We're no longer interested in, here's a brain map. We're interested in whether that brain map relates to some disorder somewhere or to some behavior in some other way. And we need a cultural shift that spans both technological consolidation. We kind of keep exploding technologies. We need to consolidate it in some ways, especially around software and pipelines, which means scientific thinking around those. Training and education, which an event like this helps with. There's a lot to learn, and there's lots of things that are changing daily. And I would say collaboration is one of the keys to this, which is at the level of complexity that we are dealing with, we can't be walking the route alone. It would be great if we could do things together. And over the last 7 years, as Ariel said, there have been several things at this event which has kind of spurned off projects that people continued post this event. So as you work on your hackathon next week, I'm sure many of those projects will kind of continue, and some of them have become papers in various places, and others have kind of continued as a collaborative team post this thing. So I wish you all the best for the next week, and I'm happy to take any questions. Okay, that was too disappointing a talk, I think. So, yes.
So we have some restrictions about the training data, even though it's basic or minimized data, we cannot serve. And I wonder if you have problems as well with some data and how to overcome that, even if you cannot consider raw data or because there's group level analysis and stuff like that.
Okay. So I'll repeat the question. The question was about we did not have consent, or we did not have appropriate consent to share the data, and hence we are restricted from using the data. Are there examples that I have that kind of relate to that story, and what can you share potentially around this? So I'll give a couple of examples around this. There is a data set, in fact, that voice and depression data set is one of them, where when we started collecting data, we did not have an open language about sharing of data. So there are a few participants in that data set which did not explicitly consent to sharing of the data. Now, the data we are sharing is completely anonymized in the sense of it's defaced, it doesn't have, but our IRB asked us to go back and contact each of those people. And this is now almost 7 years past that data set when it was collected. So not everybody is around to respond to a request like that. So we would be sharing the parts of the data set that we can share. Anybody who kind of reconsents to that, we will share and simply say, there was 5 people or 10 people that we cannot share because they did not explicitly agree to it, even though from a certain standpoint, it's de-identified data because we can remove certain features from it. But IRB asked us to go through this in this specific case because it's sensitive data. It has information about depression and suicidality and other things associated with that data set. So if you were just sharing the brain images, they might have allowed us to share all of the data because it would fall under the identified things. But because we are sharing other kind of clinical pieces, they said, hold back. And this is what has made us kind of change our consent process completely. And we ask for explicit consent anytime. And we will simply say, they have the right to say, do not share. And they have the right to ask, allow us to share. And we respect the participant's right in doing so. In the case of something that you have collected, you are governed by those rules. And you'll have to talk to your institutional resource board. If you have the possibility of recontacting, you could change the consent and ask them more explicitly whether things, and then depending on where you are, so the EU and the GDPR has very specific rules governing shareability of things and where you can share it. And I'm sure Melanie knows a lot more about this and she can talk about it if you ask her. But it really, I think, is this notion of explaining to people what will be done with their data or what could be done with their data, knowing that you can't predict ahead of time all the things that could be done with the data. So you have to assess the risk of sharing the data with the benefits that it provides. And that's something that I think has to be communicated with the people providing the data. And so in your case, I would suggest if you can recontact, open it up to them, explain it to them, and then do so. So for these future studies which are requiring data to be shared, all of that would have to happen as part of that consent process. And so for a data set that we collected, it's called the Boston Adolescent Neuroimaging of Depression and Anxiety, which is 205 adolescents imaging data, HCP style protocol that'll be available through the NDA. They all kind of consented. Well, because they were kids, they assented, their parents consented to putting this data in the NDA, and it'll be available in the NDA. So it's very important that these processes happen prior to data collection as much as possible than post data collection. Yes.
Particular group of people, especially in the local population, that will not consent, as your data will be always viable.
Fantastic point. Yes. So, and I think this is where some of these programs like Bridge to AI are going to develop newer kind of consenting processes and protection processes so that you can get at the distribution that you're looking at. I would say all of our data are biased at this point in time. Yes. But it's a fantastic point that you make, that any process that we introduce is causing bias in certain ways. I hope universal sampling comes into play, but almost nothing is universally sampled the way we might want it to.
One thing I have in mind is like, you might have heard of brain light that yeah, like it's like for those who don't know, it's a cloud-based computational. If you put your data on, it's on software. Mega expansion of that, and like people solutions to a lot of the problems.
Sure. So the question is whether to compute or not compute, and whether brainlife.io, which is a platform that allows people to run computation on data sets that are stored in BrainLife, for doing this. I would say there are a few pieces to that model, and both Anita and I know Franco and Soichi are thinking about a bunch of these things, which is when you come to BrainLife, it would be great to know all the analysis somebody else has done, right? So it's a findability part of it. And when you look at that, then the question should go through your mind, do I need to recompute? And why am I recomputing? We love to recompute, right? fMRI prep changes a patch version from point, something dot something dot x to something dot something dot x plus 2, and we want to run x plus 2, because it's easier for us to think about that than to think about what that impact could be on our data. Yes. Yes. The question is, can I step back to the previous question for a second, and then I'll answer that question. So in the notion of something like BrainLife, I think it's a fantastic model. There are kind of challenges with it, and where the community has to figure out what version changes are relevant to recomputing and why. And this was Pierre Belek who kind of spearheaded some of the work on the fMRI prep long-term stable version, was with this idea. It's like, when do I need to shift my processing and why? And so part of this is the challenge of understanding, not just that a new version was released, but what the changes are and how it might impact your analysis. And that's, I think, a good question to ask yourself, is because it will help you understand whether you understand enough about the processing or you're treating it completely as a black box. So those are things that I think have an educational component, but I think those are some of the challenges in the community in terms of who gets to recompute and when. Now, coming back to Noah's Slack question, which is, how do I find these resources? So we just heard brainlife.io. I had mentioned protocols.io earlier. It's a challenging problem. I thought Twitter had solved it for us, and Tal, as the former organizer of NeuroHackademy, perhaps should solve it for us. He's now at Twitter. And I think we live in echo chambers in various ways. And that's what makes it challenging, because we get to hear of things within. So you're here in some echo chamber, you get to hear. But maybe what you do is, when you go back, you can run a local workshop and tell all the other people around it. And hopefully information can that lens. That being said, like Nitric, there are places like Neurostars, which you can use to ask questions. So I would start with the idea of a question. It's like, I want to do X. Are there resources for it? The challenge with many of these things is that these resources change over time. So something might exist today, 5 years from now, that resource may not exist. And so this is more like a living system or ecosystem of tools that come into play. And so I would say ask. And if you want to find information, the Neuroinformatics Tools and Resources Clearinghouse, which is what NITRC is, NITRC, is one place where you can go and look up tools. The other is Neurostars, is a community Q&A forum that's run by the INCF, and that's another place you can look up the things. And I would say it's the same fair problem that we were just talking about with data, that we're now talking about for resources related to the kinds of things that we are interested in. We haven't solved it. FAIR is still a problem for resources. And it's not just finding it, it's knowing what it does well, or what you might use it for. So it has to come with enough annotation and richness for you to utilize it in some ways. There was a time when at many of these hackathons, we'd have people come up and talk about things that they ran into or they used for some of the things they do. So maybe next week you could spend 15 minutes each day where somebody comes up and mentions things that they have found useful. And then you can disseminate it, whether on Twitter, whether on Neurostars or other places. And that's how you get access to some of these resources. But I would say, if you have a question, ask. That's the fastest way to get to know if something exists that does X.